I'm Maya Takashima, and this is Wren and we did the Cas9 enzyme.
The Cas9 enzyme, also known as the CRISPR enzyme, is a bacterial RNA-guided endonuclease. Basically what it does is cleave mutated DNA. Cas9 works by using base pairing to recognize and cleave target DNA with complementary to the guide RNA. It's used in vitro transcription of guided RNA and can only cleave at a given locus if the gRNA spacer sequence shares sufficient homology with the target DNA. And what that means is that the gRNA strand must match up with the target DNA strand in order for the enzyme to cleave.
So Cas9 itself is naturally derived from the Streptococcus pyogenesis, which is an aerotolerant bacterium. Cas9 has 6 different domains, the REC1, REC2, bridge helix, PAM interacting, H&H, and RUBC. Cas9 also utilizes a cofactor of magnesium 2+, and Cas9 belongs to the enzyme class of hydrolase, which is shown by the enzyme commission number.
Each of the 6 domains in the Cas9 enzyme have a specific function. The first 2, REC1 and REC2, are responsible for binding the enzyme to the guide RNA strand. The next domain is the bridge helix, and that is responsible for aiding in the initiation of cleavage activity for when the binding of the enzyme to the target DNA strand is occurring. The protospacer adjacent motif, also known as the PAM interacting domain, initiates binding to the target DNA. And HNH and RUBC are nuclease complexes that allow the enzyme to cleave the DNA strand. HNH is responsible for cleaving the target DNA strand, and RUBC is responsible for cleaving the non-target DNA strand.
So the Cas9 mechanism is as follows. First, the Cas9 binds to the guide RNA, which is specific for a given gene. This directs Cas9 where to make the cut in the DNA. Cas9 gRNA then scans the DNA looking for a target recognition sequence, which is 2 G bases next to each other. It then, once it finds this recognition sequence, Cas9 opens the DNA double helix to check the adjacent sequence is complementary to the gRNA. If it is, and the DNA matches, the Cas9 will make the cut. Cas9 will repair it in either 2 different ways. Either the Cas9 will directly stick the ends together, creating a mutation at the point of break, or it will repair the DNA with a matching DNA sequence containing desired genetic alterations. This is how scientists are able to make any genetic modification that they desire. Here, using primer blast, this is a graphical view of primers. There are 10 different ones. These are PCR primers that were designed using the Cas9 cDNA to use in PCR amplification.
Here we use bioinformatics to develop a crystal structure of Cas9. In the crystal structure here, you can see the 6 domains that we talked about earlier, and even some smaller subdomains or less important domains that can be visualized in the crystal structure of Cas9.
And then here, using antigenic peptide prediction, this is another graphical representation of 52 different antigenic determinants. These 52 different antigenic determinants are points in the sequence of Cas9 that are where an antibody can bind.
Upon further investigation of this enzyme, we looked for some experiments that evaluated how well this enzyme worked in living organisms. One was titled Genome Editing with RNA-Guided Cas9 Nuclease in Zebrafish and Embryos. And what this group of scientists studied was how efficient Cas9 and gRNA complexes were in different known genes of zebrafish. So what this study looked at a couple different components that incorporated the Cas9 enzyme. First, they looked at how the enzyme and the gRNA complex induced non-homologous end joining in human embryonic kidney cells. They also looked at how the complex induced homology-directed repair also in human embryonic kidney cells. Then they looked at how Cas9 and gRNA induced indels in ET-SRP locus. An indel is a small insertion or deletion in the genome of a living organism, and ET-SRP is a very well characterized gene in vascular vascular endothelial development. And then finally, they looked at how Cas9 and gRNA induced indels in GATA5 and GATA4 locus. GATA5 and GATA4 locus are essential genes to endoderm and heart development.
So to do this, they first had to prepare the Cas9 complex. They converted the bacterial Cas9 cDNA into humanized Cas9 cDNA. They then, while doing this, included a nuclear localization signal at both ends of the Cas9 strand. This allowed for the Cas9 to enter into the nuclei of mammalian cells. They then fused cRNA, which is CRISPR RNA, with normally trans-encoded tracer RNA to give the gRNA. The gRNA is designed to target genome sequence at the protospacer adjacent motif. The Cas9, which is guided by the gRNA, unwinds and cleaves the double-stranded DNA of chromosomes in a sequence-specific manner as described in the mechanism previously.
So, as mentioned previously, the non-homologous end joining in human embryonic kidney cells efficiency was measured with Surveyor, of course, in the presence of the Cas9 enzyme. And Surveyor is an enzyme assay, which is an enzyme mismatching assay that detects single base mismatches, small insertions, and small deletions. And from this, they had a result that there was a 29% efficiency in the non-homologous end joining in the presence of the Cas9 enzyme. Next, they looked at homology-detected repair, also in the human embryonic kidney cells. They introduced the Cas9 gRNA and donor DNA fragments into the cell. They identified the Cas9-mediated conversion of mutant EGFP into wild-type EGFP fluorescence. They quantified this by counting the EGFP-positive cells through fluorescent-activated cell sorting. They then confirmed this conversion with Sanger sequencing of positive cells, and they found a result of 2.51% homology-detected repair efficiency.
So, encouraged by the efficiency of the Cas9 gRNA complex, they then did Cas9 gRNA-induced indels in the ETSRP locus. To do this, they designed the gRNA so it targeted the ETSRP locus. They were able to see the efficiency of that with luciferase single-strand annealing SSA recombination assay. They found that it efficiently generated double-strand breaks and homologous recombination repairs. And so they then microinjected the Cas9 mRNA and gRNA into one-cell embryos of zebrafish. Then at 50 hours post-fertilization, they randomly picked 8 different embryos. They then pulled their genomic DNA to use for PCR amplification to then conduct Surveyor and Sanger sequencing. The results of this showed with Surveyor that there was a 31.2 to 38.4% indel frequency. Sanger sequencing also confirmed site-specific indels, and they also found that over 38% of the Cas9 gRNA-treated embryos had defects in sprouting of intersegmental vessels. This is similar to the ETSRP Y11 mutant embryo at 50 hours post-fertilization. They were able to conclude that Cas9 gRNA catalyzes site-specific double-stranded breaks in highly efficient and sequence-specific manner. So then, following this, they did Cas9 gRNA-induced indels in the GATA5 and GATA4 loci. They designed the gRNA to target the GATA5 and GATA4. They were able to confirm the efficiency of it with sequence-specific cleavage in SSA recombinant assay. They then microinjected the Cas9 mRNA and gRNA into one-cell embryos of zebrafish. They used Surveyor and Sanger sequencing to look at the results. Surveyor found that at 50 hours post-fertilization, about 26.4% indel frequency occurred. And with Sanger sequencing, they saw that 50 to 90% were site-specific indels. They also found that with 24% of the Cas9 gRNA-treated embryos had cardia bifida, and this showed that Cas9 gRNA was efficient in biallelic conversions of GATA5.
In conclusion, this experiment found that the Cas9 and gRNA nuclease complex efficiently generates site-specific cleavage at about 35%, and this, of course, is when the enzyme is accompanied by other indels like the ETSRP, GATA4, and GATA5. They also found that Cas9 and the gRNA induce biallelic conversions of ETSRP and GATA5 in resulting somatic cells at about 30% of embryos. And finally, they found that the Cas9 and gRNA complex generates double-stranded breaks to induce homology-directed repair at or near the break site. These are all of the references that we use to collect this information. And in conclusion, we would just like to say that the Cas9 enzyme is a very important enzyme, and it will continue to be studied, especially as the genetic engineering field continues to progress, and hopefully soon we'll be able to look at more genetic modifications and gene altering using this enzyme.
Great. Moment of truth. 11.